Hello everyone, I'm Dr. Karthik Bharadwaj. I'm a clinical geneticist working at CSIR CCMB, Hyderabad, India. At the outset, I would like to thank Alka and the team from Bionano for inviting me to share my experience on using optical genome mapping, specifically in rare genetic disease research in Indian scenario. Um, um, in this talk, I would briefly take the audience through introduction for the uninitiated, speaking about the technology and how it works, and then describe our experience in various case scenarios like FSHD, translocations, and then cryptic SVs, highlighting the utility of Bionano in solving these complex genetic variations. And at the end, we'll briefly talk about the reference data set that we are generating for structural variants in our population. So structural variations, as you must be knowing, are an important cause of genetic diseases. And the traditional methods for detection of structural variants have several limitations. Like karyotyping has a lower resolution, though it can detect large, visible, balanced and unbalanced structural variations. On the other hand, chromosomal microarray, which has improved resolution, but it cannot detect balanced translocation. FISH is a targeted approach only where few loci can be assigned at a single time. Optical genome mapping, on the other hand, has several advantages. One being automated and has a very high sensitivity in detecting CNVs, greater than 500 base pairs for de novo assembly, and various structural variations like inversions, duplications, and translocations. And therefore, it is able to detect all classes of variants apart from SNVs. The way it works is that the major important thing is the extraction of ultra-high molecular weight DNA, and the enzyme actually recognizes the hexanucleotide sequence, which is randomly dispersed across the human genome, and labels it with a fluorescent dye. And the machine actually, once the labeled DNA is loaded onto the chip, it passes through nano channels, and the machine captures these images. And these images are further converted into digital molecules and consensus maps are generated. And this is how the real-time image taken by the machine looks like. And once these digital images or consensus maps are compared to the reference, you can make conclusions or derive different presence or absence of different structural variations in the sample. So the pattern of the just marker. So the pattern of the labels determines where the DNA molecule is coming from. And then say, for example, in this case, there are additional molecules and increased space between adjacent loci, suggests the presence of an insertion. Similarly, when some of these labels are lost in between known loci, then you can infer the presence of a deletion. And in inversion, you get a complete, physically transformation in the labeling pattern. So in the same way, you can detect other structural variations like translocations, tandem duplications, and repeat expansions. So now that we have understood how Bionano works, let me share our experience using follicle scenarios. One of the primary reasons that we got the Bionano optical genome mapping in our lab was to establish a cohort of facioscapulohumeral muscular dystrophy. It is probably considered as one of the commoner muscular dystrophies in the Caucasian population. But in India, we do not know the exact estimates of the prevalence because of lack of availability of testing or any test which could help establish the molecular diagnosis in these patients. It's an autosomal dominant myopathy and has several interesting features which makes it distinct from other muscular dystrophies like asymmetric muscle involvement and certain clinical features like scapular winging and all. But more importantly, it is the only known disease which is caused because of a macrosatellite repeat contraction. And the standard of care has been Southern blot for a very long time. But after OGM has come, it has enabled easier detection of facioscapulomuscular dystrophy. So mechanistically, this is caused by a macrosatellite contraction in chromosome 4 in a place called D4Z4 array. The normal repeat size here is greater than 10 repeat units. And when the repeats become less than 10 repeat units, And with the presence of permissive allele, the gene called DUX4, which is embryonically silenced, gets expressed, and this results in myotoxicity. In the interest of time, we'll not go further into the mechanisms of the disease, but this is how the image from the software looks like when you are analyzing FSHD. Here you can see a shorter repeat segment and the 4q haplotype, and this corresponds to about 3 repeats, while the image below corresponds to 28 repeats, suggesting that the case has likely contraction in the D4Z4 locus, resulting in the phenotype. So in our experience, what we were able to see is that the repeat count in FSHD cases is coming to about 4 repeat units, mean median repeat count, which is right in between the Caucasian populations and the East Asian populations, where in Europe the mean repeat count is 5.8, and then in Korea and Japan it is closer to 3. So this suggests population-based, epigenetic or genetic modifiers, which regulate the phenotype in these cases. And then we are in our lab are working on further to identify these genetic modifiers. And other use case scenarios include one of the first cases that we have done was in a case where there was additional chromosomal material absorbed on chromosome 12. And using optical genome mapping, we could identify that it's actually a duplicated segment from Q arm, which has been inserted into the P arm of chromosome 12. So this image shows the fusion, whereas this image, you can see the copy number variation. Right. And in another case, where there was a balanced translocation in a child with global developmental delay and autism spectrum disorder, this balanced translocation between chromosomes 7 and 21 was identified, but apparently CMA chromosome microarray was normal. So therefore, we were thinking in terms of disruption of some gene at that locus, which could be responsible for that phenotype. So initially, when we did optical genome mapping then we could not find any structural variants which could be responsible for the phenotype in the child. However, when we used T2T reference, once it became available, we were able to identify this translocation which was evident on karyotype, largely because the confidence of this translocation call improved. And further, on looking at this breakpoint, we could also identify disruption of AUTS2 gene, which is a known cause of autism spectrum disorder. And in another case where hemophilia A was falsely, inversion of intron 22 was falsely missed by a lab, again using T2T reference genome, we were able to identify the inversion. And in a short case series, we also tried to explore the utility of optical genome mapping in identifying cryptic structural variations. So because these cryptic structural variations, when they are present in the parents who are apparently asymptomatic, but can have several problems in their progeny. So they can like recurrent spontaneous abortions or birth defects. And some of them are also causative of subfertility or infertility. So therefore, it makes an important thing to identify these cryptic structural variations, which determine the recurrence risk of these birth defects or structural variations in the progeny of such couple. So optical genome mapping is, because of its technical advantages, is rightly placed to solve this kind of a problem. So in this cohort, let me take you through a couple of cases before highlighting the findings that we found in this case series. So in a 28-year-old woman born to second-degree consanguineous parents and with subfertility and first child with global developmental delay, a chromosomal microarray in that developmental delayed child identified a duplication in chromosome 11, right? And then karyotyping done subsequently actually identified a translocation between chromosome 1 and 12, both in the mother and the proband, but no additional abnormality. So you can see the discrepancy here. The CMA is suggestive of duplication on chromosome 11, and there is apparent balance translocation between chromosome 1 and 12. on karyotyping. And once we did OGM for the proband and the parents, what we were able to see is that mother was actually carrying a complex translocation involving chromosome 1, 11, and 12. And similarly, proband was also having this complex translocation. In addition to this complex translocation, as picked up in chromosomal microarray, he was also carrying a duplication in chromosome 11. Right. So then we kind of try to stitch the findings from OGM together and try to postulate what was the exact complex translocation that was happening. And because chromosome 11 was involved in both, we did whole chromosome paint of 11, whole chromosome paint with chromosome 11 related probes. And there you can identify a part of chromosome 11 being inserted in chromosome 12. So the hypothesis was that mother is carrier of this complex translocation, where you have chromosome material from chromosome 12 getting translocated to chromosome 1, 11 to chromosome 12, and also 1 to chromosome 12. So therefore, in the proband, when the proband has inherited, and of course mother has the deletion in chromosome 11, which is when the normal chromosome 11 had come from the mother along with normal chromosome from the father, it resulted in a net negative excess of material from chromosome 11, which is manifesting as a duplication and further global developmental delay in the child. So this significantly altered the recurrence risk in this couple. This is additional fish experiments using whole chromosome paint or multicolor fish, multicolor banding to kind of confirm the hypothesis that we had on the complex translocation. In another case, with developmental delay, epilepsy and hypotonia, and floppiness in infant, whole exome sequencing identified a deletion involving SLC2A1 gene, which results in GLUT1 deficiency syndrome. And then prenatal was done in the next pregnancy of the couple, where chromosomal microarray actually identified duplication of the same region. And then karyotypes of the parents was normal. Here, because with deletion in one of the progeny and duplication in one of the progeny, we were thinking of some structural variation, cryptic structural variation present in the parents, and therefore proceeded to perform OGM. And then in optical genome mapping, we could identify the deletion in the proband. And in the father also, we could see the deletion, but there was no CNV loss. To our surprise, we also identified two translocations involving chromosome 1 and chromosome 5, right? And these are actually orbiting the CNV loss. So therefore, we concluded that, and of course, on manual inspection also, we can find the deleted elements or inserted elements which are being picked up on chromosome 1 and 5. And then we concluded that in the father, a segment of chromosome 1 got translocated to chromosome 5, and this segment carries the SLC2A gene, which results in GLUT1 deficiency syndrome. And when the deleted allele gets inherited, you have a deletion, whereas if the normal copy gets inherited, you have duplication in the proband. So in rest of the cases, similarly we could identify either the structural variant could be resolved in a better manner, where additional translocation or gain in addition to the loss that has been identified using a standard of care test resulted in better resolution of the causative variant. And in some cases, it also, like the cases discussed previously, altered the recurrence risk in the progeny. So from this case series, we could conclude that in a significant number of cases, the SVs were resolved more accurately using OGM. And in about 4 out of 8 cases, the findings also resulted in altered recurrence risk. So I believe that the cases that I have discussed in the last few slides demonstrate the complete set of utility and advantages of optical genome mapping in rare disease research. But some limitations that we experienced are the requirements of fresh or frozen samples for extracting ultra-high molecular weight DNA. This prohibits us from utilizing previously banked samples, especially if only DNA is available for optical genome mapping. And certain types of SVs are poorly labeled, especially in poorly labeled regions are missed. For example, OGM could not pick up the 1 to 12 translocation in case 1. And then some smaller SVs could not be detected, for which long read sequencing is probably better. And another important limitation is lack of adequate reference data. Over the last 2 years, we've also seen the bioinformatic processing getting improved, and then the interface, user interface being improved. So evolving technology means some learning curve, and of course, costs involved could be limitation for some people. So one of the things that we are addressing in the limitations is generating reference data, population-specific reference data. We have collected samples across the country, and along with long-read sequencing and using optical genome mapping, we are generating reference data sets for structural variations in our population. And then we hope to make this data public as soon as possible. With this, I would like to end presentation. Before that, I would like to thank Dr. Divya Tej Saupati, who's my partner in crime for most of the work that I do at my lab. And Sophia Kanakavalli and Haricharan have been the primary contributors for the work that I presented today. And Dr. Shagun and Dr. Vishnu are the primary collaborators from whom the cases were sourced. So thank you.